If you remember, Mendel had derived the first law, or his first law of inheritance, which he called the principle of segregation, equal segregation. And basically what that said is if you're looking at any given character in an individual, those characters are determined by a pair of traits that individuals inherit from their parents. And when that individual is getting ready to make gametes of its own, those traits will be segregated with equal probability, hence the name, into each of the gametes, right? So we spent some time discussing the peas that could be either yellow or green, with yellow being fully dominant over green. And for the purpose of today's example, we'll discuss a second character that he had studied that was among those 7 that we discussed before, and that was the type of pea skin. So some plants would produce smooth peas and some would produce rough peas, and through these control back crosses, he could generate 2 pure breeding lines, one of which would produce always smooth peas, and the other always wrinkled peas. He would cross them to one another. The entirety of the F1 would have smooth peas, which indicated to him that smooth dominated fully over wrinkled. And then in the F2, if he were to self-fertilize any of the F1 plants, in the F2 he would get the classical 3-to-1 phenotypic distribution of dominant to recessive, okay? So he could explain very well the behavior of traits for any given character in isolation. The question then became, how do characters behave, or traits for each of the characters behave with respect to one another? And now we know a little better, but at the time you could imagine this was, if you will, logical or reasonable. One might think, for instance, that for whatever unknown biological reason, all dominant traits belong together and all recessive traits belong together. Maybe there was some sort of master regulatory mechanism by which an organism would express or place all of the dominant traits together into gametes and all the recessive ones together into gametes. So the point is that he didn't know if this might be the case. The alternative, which is the one that he wanted to explore more precisely, was that each of the characters behaved independently from one another, right? So the idea here was, why not let's make some pure breeding lines, but this time pure breeding for 2 traits at once. So through back crossing, through control back crossing, he generated a line, a pure breeding line that expressed both recessive traits. These plants produced peas that were green, that was recessive to yellow, and wrinkled, that was recessive to smooth. And then he produced plants that were pure breeding, but for the dominant traits, yellow and smooth. When he crossed these plants to one another, he obtained an F1 that was 100% yellow and smooth. 100% of the progeny was expressing both dominant traits. That perhaps was not in and itself that surprising. He knew for each character the dominant would dominate over the recessive, so here he was working with 2 dominants dominating over 2 recessives. So far, so good. The question was, what will happen when we self-fertilize, or back-cross in a way, self-fertilize, that's the more proper term, F1 plants to themselves, right? And just like we call crosses between individuals that are hybrid for one character monohybrid, hybrid for one, we call these types of crosses dihybrid crosses. These are individuals that are hybrid or heterozygotes, as we would say nowadays, for 2 characters. Yeah. Could you repeat that? Yeah, that we call any cross that involves individuals that are hybrid for one character, or as we would say in modern terminology, heterozygote for one locus, monohybrid crosses. They're hybrid for one, mono. These crosses that involve 2 characters that are hybrid, we call dihybrid. And should they involve 3, trihybrid, tetrahybrid, pentahybrid, and so forth, right? So here's where the modeling type of thinking came in, the sort of the mathematical type of thinking. We in a way, went through Mendel's journey from observation to hypothesis to model or hypothesis testing last week. Now we can imagine that he was in a mindset that he could treat the behavior of each character mathematically and approach the question with math. So if one postulates that each of these characters will behave independently, then looking at a dihybrid cross is like looking at two monohybrid crosses that are happening simultaneously. We will revisit this concept today, but the idea here is if P skin and P color behave independently from one another, I'll treat them independently. And the question of what type of progeny, or what is the probability of obtaining any given type of progeny, I can calculate using math, using that famous rule of multiplication, by which I can multiply the probabilities of two independent events that I'm asking the probability that they happen simultaneously. Okay? So let's say, for example, that in this dihybrid cross, he was interested in predicting what would be the probability of plants producing wrinkled and yellow peas. Because there is an and condition here, what he would do is calculate the probability of wrinkle being the recessive. The 3-to-1 would indicate that there was only 1/4 of the progeny that would show that recessive trait. And yellow being the dominant, there would be the 3/4 probability that there would be that dominant trait. So anytime that from a monohybrid you're looking at the dominant trait, you expect a 3/4 probability. Anytime you're looking at the recessive, you're expecting 1/4, that famous 3-to-1. I'll say that again because I don't know if we emphasized it enough last week, but when we say 3-to-1 phenotypic ratio, another way of expressing that is 3/4 to 1/4, because that 3-to-1 is based on a total of 4, right? Cool. So mathematically, one might predict that the probability of wrinkle and yellow equals the probability of wrinkle times the probability of yellow, and he could calculate that and get a number 3/16. So he went to, instead of going to town, he went to the garden and started setting up all of these crosses, and sure enough, the data supported this, right? The prediction was that about 3/16 of the plants in the F2 progeny would have wrinkled peas that were yellow, and that's what he saw experimentally. Moreover, one could take the same approach, and instead of asking about the probability of any specific combination, one might ask about the entire distribution of combinations. And again, when approaching this, when asking about the entire distribution of all possible combinations, we use a binomial expression, right? So if we would like to know the probability of either round and yellow, or wrinkled and yellow, or round and green, or yellow and green, we're basically using the FOIL method that you would use to solve one of these binomials, right? So in other words, if the probability of round or wrinkle from that monohybrid is 3/4, and the probability of wrinkle is 1/4, together they will make up the probability of round or wrinkled. And we multiply that by the probability of the plants producing either yellow or green peas, which distribute to 3/4 plus 1/4. We solve this, and we get the entire distribution of 9/16 of the F2 progeny will have round peas that are yellow. 3/16 will be round, a dominant trait, but green, the recessive. Another 3/16 would be wrinkled, the first recessive, but yellow, the second dominant, and then only 1/16 of them would have both recessive traits. All from math. But then he would go, do the corresponding crosses, essentially let a whole bunch of F1 self-fertilize, count the plants, count each type for these possible combinations of traits, and the numbers matched. Okay? So this was, if you will, is coming full circle in that he's going from a model of how things could work, generate a prediction, and then match that prediction to experimental data. Right? There was no prior observation. He could start right away with some mathematical formulae. Any questions? No? All of this just to say that his conclusion became his second law of inheritance, the one that we know of as the principle of independent assortment. And it basically says, I have the principles of equal segregation up there for your reference, but the principle of independent assortment, or the second law, basically says that every time that an organism sorts alleles or traits for two characters into gametes during meiosis, he didn't know about meiosis, we do, it's like this organism is tossing two coins simultaneously. All possible allelic combinations have equal probability of being sorted into a gamete. Now, that is not to say, when we say that all possible allelic combinations, that obviously depends on the allelic combination of the individual making gametes. If we have an individual, to go back to that idea of the coins with two heads or two tails, if we have an individual that is, that's right, an individual that is homozygote for both characters, either dominant or recessive. Let's say this individual, dominant, homozygote dominant for A and homozygote recessive for B. It's not like this organism will produce all possible allelic combinations because it only has dominant alleles for A and only dominant alleles for B. So the idea of all possible allelic combinations becomes a little more clear, a little easier to see when you're thinking about hybrids, when you're thinking about heterozygotes for a given character. When we're dealing with homozygotes, it's not like the second law breaks down or anything. It's just that all allelic combinations becomes something like this. Let's see. We can say this and then this. So like that, B and A, right? Where you have all possible allelic combinations if you were to follow the parent origin of these alleles. But when it comes to the actual allelic combinations, it's really only one, a dominant A and a recessive B. So let's not forget that this business of alleles sorting with equal probability into gametes applies to the actual physical alleles, the actual pieces of DNA that are going into gametes during meiosis. But if the identity of those pieces of DNA are the same, then at the end of the day, from a genetic perspective, the type of information being passed on is the same. Does that make sense? We're going to revisit this again towards the end of today's lecture, but this idea that you shouldn't confuse allelic combinations or allelic identities with gamete identity. You can produce gametes that are different from a molecular physical perspective and yet carry the same type of genetic information. Okay? Any questions now? We're good? All right. Now I have a question for you. We said that Mendel could either address the probability of specific combination of traits by using the multiplication rule, or could derive the distribution of possible phenotypic combinations based on a binomial. Now, all of that addresses what is the probability of allelic combinations and therefore phenotypes or physical manifestations of these traits in the F2 progeny. Yet, the second law is formulated from the perspective of what happens to the alleles of different characters and how they segregate independently from what alleles in other characters do into gametes. So if we were to think, or if we were to ask whether the second law is true or not from an experimental point of view, but looking at the gametes, how could you look at the alleles that go into gametes? How could you look? So the question is phrased, how can we determine the allelic compositions of gametes produced by an individual? So by saying, I'm going to look at the phenotypes of the F2 based on what I know about the phenotypic distributions, all possible combinations of phenotypes from a monohybrid cross, I expect to see that 9 to 3 to 3 to 1. But I thought you meant something slightly different, which is interesting. Why don't we look at the gametes that the F2 produces? Anytime you want to look at the gametes, or rather at the alleles that a given organism has, one way to do that is by looking at the gametes, right? And here's why. If we are dealing with diploid organisms and there is some case of full dominance, by looking at the phenotype of the organism, we can tell what the allelic composition of that organism is only if they are phenotypically recessive. Should they be phenotypically dominant, we're stuck. We can't really say with certainty what their genotype is. We can't say with certainty what their allelic combination is. If they're homozygous dominant, they would show the dominant phenotype. And if they were heterozygous, they would show the dominant phenotype as well, right? So the only way to look at alleles that an organism has is by looking at the gametes they produce, because now we've isolated those alleles from one another thanks to the first law, thanks to this idea that alleles assort themselves and separate from each other, segregate from each other during meiosis. In past semesters, I sometimes get people saying, well, can you use molecular techniques to look inside of the gametes? Yes, you can. In fact, we would probably do that nowadays, right? But back then, Mendel didn't have that, and those didn't come around for a long, long time. Okay, so the second question is, how do you look at the distribution of possible allelic combinations in gametes? Do you guys remember how we did that for the monohybrid crosses? If you recall, Mendel took that F2 that showed when he was working with only one character, let's say P color, he took that F1 that showed a 3-to-1, and then he self-crossed each of those F1 plants to obtain an F2. Am I getting that right? Yeah. No, actually, he self-crossed the F2s to look at the F3. And in the F3, if you recall, he found that the plants that show the recessive phenotype were, now in hindsight, expectedly, pure breeding recessive. So those plants that show the green peas always produce green peas. Now, when it came to the yellow pea producing plants, for about every 3 of them, 1 of them was pure breeding. It would always produce plants with yellow peas, but the other 2 were hybrid. So he established that not only is the F2, not only does the F2 follow a 3-to-1 phenotypic distribution, but it also followed a 1-to-2. The F3 followed a 1-to-2-to-1 pure breeding to hybrid to pure breeding, right? So the idea here is that conceivably, and I honestly don't know if Mendel ever did this or not, but conceivably one could say, well, if instead of worrying about phenotypes in that F2 generation, I think about them in terms of genotypes, I know that a quarter of them would be pure breeding dominant, about half of them would be hybrids, and about a quarter of them would be pure breeding recessive. That was for one character. The same applies to the other character. So if we're looking at 2 characters that are distributing alleles into gametes following the first law, when we look at the entire space of probable combinations, we again have, in this case, not a binomial, a trinomial, and then when we solve it, we get this long string of different probabilities. Once again, I don't know if Mendel did this or not, but this would have been a way to look at this problem. So is it true that after conducting self-crosses of the F2, you would get about 1/16 that are pure breeding for both dominants, 2/16 that are pure breeding for the dominant but hybrid with the second character, 1/16 that is pure breeding for the dominant and pure breeding recessive for the second, and so forth. Yeah.
This would involve self-crossing the F2s.
Correct.
And then testing how the F3s.
That's right. That's right. So you would self-cross the F2s, and then check the progeny, the F3 progeny, and if they show only one trait for one of the characters, you can sort of approach the conclusion that for that character the organism is pure breeding. But then you have to look at the other characters. So what could have conceivably happened? Let's home in on this guy here. What these two 16s would have shown is 2 out of the 16 plants in the F2, when you self-fertilize them, they would show that they would always produce the smooth peas. But then about 3/4 of those peas would be yellow, and 1/4 of those plants would produce green peas. All of them smooth, but then 3/4 yellow, 1/4 green, and so forth. Only 1/16 of them would be pure breeding for both recessives. So this is one approach. Kind of time consuming. And with plants and with any other organisms that reproduce in large numbers, doable, but time consuming. So it's a shortcut. And we did touch on this, and I said that we would come back to it, and we will come back to it again next week. But it's related to sort of an important concept that we mentioned last week called the test cross. And I don't think you have this slide here or in your decks, but you'll have it later in Canvas. If you recall, the idea of a test cross is crossing any individual of unknown genotype. You can look at it and know its phenotype, but you don't know what's its allelic composition, and then crossing it to a test individual, this is by design, that is pure breeding recessive for all of the characters that you're interested in. So in this case, we could have taken a dihybrid individual from the F1 in this second round of crosses, right? And then cross it to an individual that is pure breeding recessive for both characters. So green peas and wrinkled. And more importantly, every time that you back cross these plants, every time you'll get green peas and wrinkled. Why a test cross allows you to look at the identity of the alleles that go into gametes is because the pure breeding individual, be it for one character, 2, 3, 4, 5, will always place recessive alleles into its gametes. So when those gametes from the test individual in the cross fertilize with the mystery gametes from the mystery individual, since the test individual is providing nothing but recessives, you will be able to see what the other one is providing. Because if the other one is putting a dominant into a gamete, you will see the dominant phenotype. And if the other is putting a recessive allele into its gametes, then you will see a recessive phenotype. Right? Last week, we used it with the simplest of examples. We have a plant. In that case, we were talking about flower color, but it was a plant with the dominant trait, purple flowers, and we crossed it to a white flower plant. And the whole idea of the test cross in that case was simply asking the question, is this dominant phenotype plant, the purple flower plant, heterozygote or homozygote for the dominant allele? If we got nothing but purple flowers in the progeny, then it was a good indication that that plant must have been a pure breeding dominant or homozygote dominant. But if it was roughly half and half of purple flower plants and white flower plants, then it was an indication that that parent was heterozygote. We placed the emphasis on the genotypic identity of the parent in that case. But we did mention, though, that the test cross is a way to really probe the allelic combination of gametes, right? So, I don't know if Mendel did this either. Probably he didn't do this. The idea of a test cross came a little later on, I think. But this would be a much quicker way of determining or in a way supporting experimentally the second law. If you take a dihybrid individual, let it form its gametes and then test those gametes against a double homozygote recessive individual that makes gametes with nothing but recessive alleles in them, then you would find out the different relative proportions of the allelic combinations in those gametes produced by the F1 individual. And in that case, you would find that there is 1/4 of both dominants, yellow and smooth, 1/4 of the first dominant, yellow, and the second recessive, green. Sorry, wrinkled. I'm gonna mess it up here. Yellow. Oh, come on. Sorry. Yellow and wrinkle. Then 1/4 of the other one, green and smooth, and 1/4 of green and wrinkle. I hope this doesn't throw you off much. This can be confusing because we go from that famous 9 to 3 to 3 to 1 that we will hammer over again and again during this week and a little bit next week. But this comes handy next week when we talk about linkage and mapping and recombination, right? Because next week it will almost all be about how you use these test crosses to find out the allelic combinations that go into gametes produced by an individual. So the emphasis here is in that these 1 to 1 to 1 to 1 are not phenotypic ratios. They are allelic ratios or allelic combinations, the ratios of allelic combinations that go into the gametes that we're testing by fertilizing those gametes to gametes produced by an individual that provides nothing but recessives. Okay? So the 9 to 3 to 3 to 1 is what you get through a dihybrid cross, a cross between two individuals or a hermaphrodite that can fertilize with itself that is heterozygote or hybrid for both characters. Here we're talking about a cross between a dihybrid or a hybrid for two characters and then a double homozygote recessive. Different cross, different outcome. Okay? Cool. So, just like we did for the first law, let's take a look at how the second law can be explained in modern molecular biology or molecular genetics terms. So last week we saw how during meiosis, homologous chromosomes align across each other on both sides of the equatorial plane of the meiotic cell, and during meiosis one they separate from one another. And that explained how the two alleles for any given character separate from each other during meiosis. Then there was a second meiotic division that separated the chromatids, sister chromatids in each duplicated chromosome, but that at the end of the day, the probability that a given allele of each type would end up in a gamete was 50-50. Okay? Here what we have is a cartoon showing two pairs of homologous chromosomes. So the first one is carrying the R locus, the one that codes for P skin, and the second one is coding for the Y locus, the one that codes for P color. By the way, I should point out that the R comes from round. I keep calling it smooth because in my mind, smooth versus wrinkled is easier than round versus wrinkled, but the R comes from round. Okay? So these are the chromosomes that exist in a pre-meiotic cell, but then that cell decides that it will undergo meiosis. So first thing it will do is chromosome replication or DNA replication. Now each of these chromosomes will have a sister chromatid that has an identical copy of its corresponding allele. Right? And during meiosis 1, we have the two homologous chromosomes for the R locus facing each other on both sides of the equator, and then the two homologous chromosomes carrying the Y locus facing each other on both sides of the equator. Right? So that happens in meiosis 1, and after meiosis 1, these two, each member of each homologous pair will separate from one another, which sends the dominant R and in this case, the dominant Y to one meiotic 1 daughter cell, and the recessive R and recessive Y to the other daughter of meiosis 1. Question for you. If that's what happens in meiosis 1, why do we say that the alleles for each of these two characters segregate independently from one another? I'm showing them here, the dominants going with the dominants and the recessives going with the recessives. How can now they separate from one another and end up in a different combination, say a dominant of one with a recessive of another, and vice versa? So how could ever, in the previous slide we said that the probability of a gamete inheriting a dominant R and a recessive Y was the same as a gamete inheriting the dominant R and the dominant Y, or the probability of a dominant Y going with a recessive R and both recessive. So how can they separate after meiosis 1? What a pickle, right? Here's where it gets a little tricky, but it'll make sense. The second law of Mendel is not explained within a meiotic division. The second law of Mendel is explained when you look across meiotic divisions. Why? An individual will never produce gametes through one meiotic division. A plant will never make exactly 4 pollen grains and 4 ovules. A mammal will not make 4 sperm cells and 4 eggs, and so forth, right? Individuals make lots of gametes through lots of meiotic divisions. And the way we explain Mendel's second law is by taking into account that the orientation of each of these pairs of homologous chromosomes is random relative to each other. So in about half of the meiosis, both chromosomes with the dominant alleles in them will end up on the same half of the cell. And the chromosomes with the recessive ones will end up on the other side. But in the other half of meiotic divisions, it will be the other way around. It will be such that we're looking at it from the point of view of the chromosome for the Y locus, but it would be the same if we thought of it in terms of the R chromosome, its relative distributions between them. So they've inverted, right? So now in this silly cartoon, the purple is now to the left and the yellow to the right. And I'm talking about the color of these chromosomes in the cartoon. The recessive ends up to the left and the dominant to the to the right. Did that make sense? So the equal segregation, the first law can be explained within a meiotic division because every meiotic division will separate the alleles for each locus. But the principle of independent assortment can only be explained if we look at all meiotic divisions together. And when we consider that about half of them will show one relative orientation of the chromosomes, and then the other half will show the other. Now, here we're playing around with 2 chromosomes, and there is 2 possible orientations. If we were looking at all the chromosomes in this plant, and my understanding is that this plant has 7 chromosomes, that's its haploid, or its haploid number, yeah. Then we would be looking at 2 to the power of 7 possible combinations, relative combinations of the chromosomes, right? Does that kind of make sense? If you fix the first chromosome, then the second chromosome can be one way or the other. That splits the space of probabilities into 2. Then the third chromosome can be one way or the other, and then one way or the other again, splitting into 4, 8, 16, and so forth. Yeah? So for only 2 characters, there is 2 possible relative distributions of homolog chromosomes across the equatorial plate, and therefore that's how we explain the 1-to-1-to-1 relative distribution of allelic combinations across the gametes. Okay? Now, you'll notice that this was, then of course, all of it would be followed by anaphase 2, right? Meiosis 2, concluding with anaphase 2, which separates the chromatids within each chromosome, such that each gamete ends up with one chromatid carrying each of the alleles. Again, we go back to this idea of, whoa, hold on a second. So where is the 1 to 1 to 1 to 1? Now we have 8 gametes. Yeah, but when you look at the details, it turns out that these two are identical to each other, these two are identical to each other, these two to each other, and so forth. Which means that from a relative proportions point of view, it is equal probabilities of each of these combinations giving rise to the 1 to 1 to 1, or 25% each. Now, what's missing in all of this? What's missing in this meiosis? Kind of mentioned it before. The crossing over, right? So what does crossing over do to Mendel's second law? Let's take a look. When crossovers happen before meiosis 1, or before completion of meiosis 1, I should say, now what is opposing each other on both sides of that equatorial plane for the premeiotic 1 cell are not two chromosomes with two pairs of identical chromatids anymore. They've swapped information, so now you have one original chromatid, one that is a little bit, has a little bit of the other, of the homologous chromosome, vice versa, and then the original of the second homolog, right? So you end up with this combination of weird chromatids. But much like what we saw last week, you have up here what the outcome of chromosomal and, in a way, genetic composition of the different gametes would be, right? And then down here, I reproduce for your reference what you expect to see if there was no crossovers. So is the outcome with crossovers the same as the outcome without crossovers? I see some no's and some yes. So who's for no? Why are they not the same? Different combinations, right? Different colors, we can see it, right? Different. So those gametes are different. But those who said yes, why did you say yes? I know you said yes.
Well, maybe it's like depending on which region of the chromosome like had that crossing over of it.
Uh-huh.
Maybe the specific alleles weren't swapped, so it stays like the same. But it depends on if crossover is different in that region.
But if you're saying if it depends on whether or not crossover happened, so we go back to what we're discussing last time. This is just the same with an extra character added to it. Yeah.
Following up on what she said, could it also be that what was exchanged doesn't necessarily affect the allele?
Oh, now I think now I understand. So the argument is, well, what if the crossover didn't affect the loci in question? But from this cartoon, you can see that it did, right? So the question is, are these gametes on top different from the ones in the bottom? One answer was, yes, they are. Look at them. The color pattern is totally different, right? Here you have 8 gametes, but in reality what you have is 4 pairs of identical ones. Here you have 8 gametes that are completely independent or different from one another. That is true. What is also true, though, is that crossing over did not affect the second law when it comes to the assortment of alleles for these two characters. Why? Here's why. Those arrows there show that for each of the gametes with crossover, there is a corresponding gamete that has the same allelic combination without it. So what crossover does, once again, is change the molecular identity of the gametes being produced by the organism. With crossover, no two gametes will be the same, right? Without crossover, you have pairs of gametes that are identical to each other. But with crossover, no two gametes will be the same. And I'm talking the millions of gametes that an individual produces, since there is several crossover events through our chromosomes, no two gametes will be the same. Yet, from the point of view of what happens, what is the fate of each of these alleles from two separate characters, they still follow Mendel's second law. They will distribute into gametes independently from one another, and each of them will have equal probability of ending in a gamete, which means that all allelic combinations have equal probability, or equal chance of happening. Did that make sense? Yeah? One way to approach this question of, well, but what happens if there is crossover? What happens to Mendel's laws if there is crossover? We did it last week. What happens to Mendel's first law if there is crossing over? And then we concluded that nothing happens. And then today what we're doing is ask what happens to Mendel's second law if there is crossing over, and we just saw nothing really happens. Another way to sort of fully embrace that realization is to think that, guess what those plants that Mendel was working with were doing when making gametes? They were crossing over. No one knew it yet, but those plants were doing crossing over. And yet he could derive his laws by observing progeny, because crossing over does not affect the behavior of the alleles and how they behave with respect to one another for a given character and with respect to alleles from a different character, whether there is crossover or not. Things are going to change next week when we start looking at alleles for loci or characters that live in the same chromosome. But we're going to leave that for next week. Okay, so let's work through some examples on how you approach solving crosses that involve two loci. Like we did last week, let's say that we're crossing these two. There is a dihybrid cross, so we're crossing two individuals that are heterozygote for these two loci. And then the question is very specific. What is the probability of obtaining an individual in progeny that is homozygote recessive for R and heterozygote for Y? So essentially what we're asking is, what is the probability that parent number 1 produces a gamete with a recessive R and a dominant Y, and parent number 2 produces a gamete with a recessive for both, right? Now, based on Mendel's second law, the probability of these allelic combinations is equal to the product of the probability of each of these alleles ending in a gamete, right? So essentially we're asking, what is the probability that the first parent puts a recessive R in one of its gametes? And I'm asking all of you, what is that probability? Louder? 50, 0.5, 1 half, right? Yeah. And what is the probability that that same parent places a dominant Y in the gamete? 50% again, yes. So what is the probability that both of these alleles end up in that particular combination? Is the product 0.25. Cool. Let's look at the second parent. What is the probability that the second parent, or this plant when making, I don't know, ovules, places a recessive R in that gamete? 50%. And what is the probability that it places a recessive Y? 50%, right? 25. So now the probability of these two gametes fertilizing one another will be the product of those probabilities, and that's 125, 0.125. What's the problem with this approach? Correct. I'm going to repeat it so that it shows up in the recording. Doesn't account for all possible combinations, and you didn't say, but I know you were thinking it, of the heterozygotes, right? So the problem is, again, like last week with the heads, because I said very loosely that to get a heterozygote in the F2, what is the probability of a heterozygote in the F2? The first parent had to provide a dominant and the second parent had to provide a recessive. But these would work just as well if the first parent provided a recessive and the second parent provided a dominant. And it is very easy to forget that that's also a combination that will give you the desired outcome, and therefore you may forget to add them up. So we just calculated that the probability of this particular route is 0.125, and this particular route is also 0.125, so you should add them up together for a probability of 0.25, right? But it is easy to forget, just like it was easy to forget that you can get heads in the progeny with one parent contributing a dominant and the other a recessive, or vice versa. So that's why approaching these things with probabilities can be tricky, can be a little tricky. Alternatively, and this works much better, we can do what Mendel did and think of a dihybrid cross as two monohybrid crosses that are happening simultaneously. So yes, we are told that the individuals being crossed are dihybrid, so heterozygote for those two loci. But if we also know that these two loci segregate independently, meaning they follow Mendel's second law, we can treat the cross as two monohybrid crosses happening simultaneously. So we divide the cross as two monohybrid crosses, and then we ask the question, what is the probability of homozygote recessive for R out of a monohybrid cross for the R locus, and what is the probability of heterozygote out of a monohybrid cross for Y? And do you guys remember what the probability for a homozygote recessive was out of a monohybrid? 1/4, right? This is 1/4. And what is the probability of a heterozygote? 1/2. Right? So that's 1/4 times 1/2. That's 1/8. It's a lot simpler. So split up. No matter how many loci you're looking at, split it up into an equal number of monohybrid crosses. It's a lot easier to remember that 1 to 2 to 1 genotypic distribution out of a monohybrid cross than remembering 1 in 16 plus 2 16s plus blah, 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 right? What if we go crazy and we ask about a cross out of 5 loci, right? Well, again, we can break it up into each of these monohybrid crosses. So in the interest of time, I don't know if we'll have time to try it. What I would invite you to do, I'm going to show you the answer, but then go back home after you haven't thought about this for a while and try for yourselves. But what is the probability of this particular genotype in the progeny? What you do is you break up this penti-loci cross into 5 monohybrid crosses, or 5 monolocus crosses, because some of them are monohybrid, some of them are not. So for instance, this here is not technically a monohybrid cross because it's one hybrid cross to a homozygote recessive and so forth, right? But you can break these penti-loci cross into 5 monolocus crosses, and then you do that, you calculate the probability of each of the desired genotypes individually, then you multiply across, because remember, what they're asking us is the probability that each of these things happens simultaneously in progeny, so we're asking about the simultaneous combination of all of them. We'll do one so that we know what we're talking about, but let's say for M, the question asks about a heterozygote M, and we know that we're crossing a heterozygote M to a homozygote recessive. So what is the probability of getting a heterozygote out of that? We can't follow the 1 to 2 to 1 because it's no longer a monohybrid cross, right? But this second parent can provide nothing but recessives, right? So what is it that we're asking of the second parent? We're asking for a dominant. And what is the probability that that second parent provides a dominant to the cross? 0.5, right? Because it's a heterozygote, so 0.5. So the first parent produces gametes with dominant alleles 50% of the times. The second parent produces gametes with recessive alleles all of the time. So the probability of both happening is 0.5 times 1 or 0.5. And then you can do the same for each of the individual crosses. So it's not about getting great at figuring out the probability of allelic combinations for any number of loci into gametes. It's more about getting great memorizing perhaps the 2 or 3 rules for one locus crosses. So the monohybrid cross would be 1 to 2 to 1. Crosses between homozygotes, let's say a homozygote dominant and homozygote recessive, will be 100% heterozygote, nothing else, right? And then there'll be different combinations. Any of these, if I had asked you what is the probability of progeny that is homozygote dominant, heterozygote for P, homozygote recessive for Q, blah, 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 blah. Right off the bat, you know that this, given these parents, is impossible, right? Because the second individual, this one here, the homozygote recessive, cannot provide a dominant to the progeny. So you cannot have a homozygote dominant because the second parent doesn't have one. So that term would be a 0 and it would negate everything else. Okay? So you just need to get really good at figuring out the probability of different outcomes from monolocus crosses, and then treat a multi-loci cross as a collection of monolocus crosses. Figure each one separately and then multiply across. Yeah?